Now that you have learned essential details about bacteria and prokaryotes in general, such as the structure, the function, the sources of genetic diversity in bacteria, it is now important that we turn our attention over to the diversity of prokaryotes. And in this slide that I am opening this mini video with, you're going to see what can be represented, or can be understood as a phylogenetic tree of life, but it's only emphasizing the domain levels of organization. So we can see the three main domains of life, beginning with the domain Eukarya, the domain Archaea, and the domain Bacteria. And so you can use information you've learned previously, like in Bio 212 regarding phylogenetic trees, and draw inferences, draw conclusions about how scientists explain the connections, the relatedness between living things as they may be sharing a similar evolutionary track or history. And so one of the interesting details about this tree is that it places two domains, domain Eukarya and domain Archaea, as sister taxa. Even though the eukaryotes are those organisms made up of eukaryotic cells where a nucleus exists, they are going to be a sister taxon of the archaea, which are prokaryotic cells. Some students are shocked about this because they would imagine that the two prokaryotic domains, domain archaea and bacteria, should be more closely related from a phylogenetic point of view, but the reality is they are not. And there are reasons for this we're going to be explaining or studying in this chapter 27 of our current edition of the textbook. And so you can see here the domain bacteria is going to have an ancestor that is not shared by those members of the domain Eukarya or Archaea. One of those domains we're going to be exploring in the next few slides is going to be precisely domain Archaea. And I wanted to give you an idea for why exactly this closer association from a phylogenetic point of view between domains Eukarya, the eukaryotic organisms, and domain Archaea, which are prokaryotic, is going to exist. Let's focus our attention, for example, when you look at RNA polymerase. And so RNA polymerase is that enzyme that is responsible for copying DNA during the process of gene expression. RNA polymerase is responsible for transcription, making a transcript of DNA in the form of messenger RNA. And so if you look at Archaea, there's going to be several kinds of RNA polymerase, and there's also going to be several kinds of RNA polymerase among eukaryotes. However, bacteria, members of domain bacteria, only have one kind. Another interesting detail that you can pick up from looking at similarities between a prokaryotic type of domain like Archaea and domain Eukarya is going to be that the start amino acid during protein synthesis for both Archaea and Eukarya is going to be methionine. And so you may remember when we were studying gene expression and the translation of genetic message in mRNA, that the first codon, the start codon, always coded for methionine. And so that's true for these two domains, Archaea and Eukarya. But if you look at bacteria, there's actually going to be a different variation of methionine, which is known as formal methionine. And so this is going to be an amino acid version that we don't see within the domains Archaea or Eukarya. Another common factor, you can call it that, between Eukarya and Archaea is going to be the presence of histones. Histones are going to be proteins that are used for organizing DNA. So the double helix is going to wrap around clusters of these histone proteins, and histones can be found in some species of Archaea. It's going to be present in all eukaryotic DNA inside of the nucleus. However, we don't see any form of histones in members of domain bacteria. And so this is just an example of a few similarities that exist between Archaea and Eukarya. Of course, there are also going to be similarities between the two prokaryotic domains. So if you look, for example, at the chromosome, we've said before that chromosomes are circular in bacteria, and they're also going to be circular in archaea, whereas eukaryotic chromosomes are going to be more of a linear fashion with caps on the ends we call telomeres. Another similarity you can find between bacteria and archaea, and this is kind of obvious, is that both of these types of cells, the cells of these organisms, don't have a nucleus, so that's why we call them prokaryotic. And a few other similarities can also be identified. But at the molecular level, there's going to be a greater number of similarities between archaea and eukarya, the eukaryotic organisms, to place these two, as we saw previously, as being sister taxa, as having, I lost my laser pointer function. Here we have it again. So as being descendants of one shared common ancestor for both of these domains. And you can see that in this slide. All right, so let's take a closer look at domain archaea. Members of domain archaea are going to be found often living in extreme environments. What I call by extreme is going to be like extreme heat, extreme salinity, places where other forms of life are not possible. Even bacteria, what you can expect to find there would be archaea type of prokaryotic organisms. So for that reason, because they live in extreme environments, we call them extremophiles. The suffix here, Philes, this means love, so like extreme loving organisms. One of those examples of an extremophile are going to be the extreme halophiles. Halo means salt. So these are going to be salt-loving archaea type of prokaryote. They are going to be found thriving, doing really well in places where the salinity is even greater than the salinity you can expect to find in the ocean. What are some of those places? Well, I can tell you a couple of them that you might have heard of. For example, there is a Dead Sea in the Middle East. There's also the Great Salt Lake in Utah, and these are places where these extreme halophiles are going to be found. They're going to be members of a clade known as Euryarchaeota. And so if we go back to the organization of domain archaea, you will see that there are 5 groups of archaea prokaryotes, and these euryarchaeotes are going to comprise a large number of the species that have been studied, named, and identified. The prefix eury simply means broad, like a wide range. And the name was given to these halophiles, the clade Euryarchaeota. Halophiles are an example of those, because these kinds of archaebacteria can be found in a broad range of conditions. They can be found in a broad range of environments as well. And so that's simply what Eury means, Euryarchaeota. There's another type of extreme type of archaebacteria, and that is going to be the thermophiles. So remember, philes means loving, and thermo here refers to heat. So these are going to be heat-loving bacteria. A lot of these thermophiles, the majority of them, are going to be placed in the clade Crenarchaeota. And I'm just going to move back a couple of slides again just to show you that here we have the Crenarchaeota. And the prefix Cren, in this case, means spring. And the reason this group of thermophiles is giving the clade name Crenarchaeota is because they are often found in places where you have these hot springs where superheated water coming from underneath the crust of the earth is projecting out. This is a scene captured from a place like Yellowstone National Park, where we have these springs with super boiling water that is at a much higher temperature than that of boiling. And in this place where it's so hot, the only thing you can expect to be found alive there is going to be species of extreme thermophile bacteria. There's another clade worth mentioning among the archaebacteria, and these are going to be the methanogens. Methanogens also have a name that describes what they do. Methano, it's making reference to methane, and gen, which means genesis or origin. These are going to be bacteria or archaebacteria that as part of their metabolism, they generate methane. And so what they usually employ is going to be carbon dioxide. That is going to be a source of carbon for making other compounds. And with carbon dioxide, they are going to be oxidizing hydrogen gas. And so with hydrogen and carbon, one of the things that can be made is going to be methane, CH4, the most simple of organic molecules. And that is going to be a waste product. Methanogens are going to be strictly anaerobic organisms, meaning they must live in complete absence of oxygen. Oxygen will indeed kill these microbes. So there you have a good sample of what these archaebacteria are going to be. Let us shift now our attention to a second domain of prokaryotic organisms, and it's the domain that has been given the name bacteria. Domain bacteria includes all those single-cell prokaryotic organisms that you can expect to find on any place on Earth where life, for us like humans, is possible. They can be found in the soil of your front yard. They can be found on the floor of your house. They can be found in your bathtubs. And they are even found living on top of us, on our skin. They can also be found inside of our bodies as well. So domain bacteria is going to be a widely diverse group of organisms found in many different parts of the world. And they perform a lot of beneficial functions for the environment. However, as I mentioned before, a few of these members of domain bacteria are responsible for diseases, some serious diseases that can even result in death if not treated. And for that reason, bacteria are often associated with a bad reputation in our minds. And I'm just hoping that I can change your mind and help you understand that the majority of bacteria living on Earth are actually going to be beneficial to the environments where bacteria live, and directly or indirectly beneficial to us as well. So what are the subdivisions of domain bacteria? And again, if you want to see a summary of those, I'm trying to, I was trying to find a shortcut for moving between slides. If you want to see the subdivision, the breakdown for the main groups, the main clades within domain bacteria, here we have Proteobacteria, Chlamydias is another clade, Spirochetes is another clade, Cyanobacteria, which we've seen in lab, and also gram-positive bacteria. So let's just go down the list and see a few examples of these bacteria, making reference to what they are and what they do. Proteobacteria are going to be all gram-negative. And so by now you understand quite well the concept of gram staining. Included in this group of the Proteobacteria is a wide variety of microbes, of bacteria. I'm sorry for the redundancy here. And I want to make this illustration a little bit larger so I can point out a few things to you. The members of domain bacteria, as you can see in the upper left part of my screen, are broken down into 5 groups known as alpha, beta, gamma, delta, and epsilon. So these are the first 5 letters of the Greek alphabet. And so if we look at alpha Proteobacteria, we're going to find here a lot of the bacteria that live in the soil. Here, Rhizobium is going to be one of those specimens we see in lab. Rhizobium is responsible for making nodules in the roots of a few plants, including members of the bean family. So plants we call legumes, members of the family Leguminosae, they are going to have often associations, their roots have associations with these beneficial bacteria, which provide a benefit to the plant by helping capture nitrogen from the soil and then make that nitrogen available to the root of the plant. In exchange, the root of a plant provides a safe environment for these bacteria to grow, and they also share some of the sugars plants make through the process of photosynthesis. So in this illustration, and I wish that I could point, maybe you can see this little hand, which is what my cursor is looking like. For some reason, my laser pointer is not functioning when I do this zoom in. These dark objects you find are going to be the bacteria. The larger round object you see right here is going to be like a cell in a plant, in the root of a plant. And so this cell is going to be part of that nodule. They look like little tiny tumors in the roots of plants. Another subgroup of proteobacteria, remember these are all gram-negative bacteria, is going to be nitrosomas. Somonas, the name is nitrosomonas. Somo means body or object, and nitro makes reference to nitrogen. These are also nitrogen-fixing bacteria. Gamma proteobacteria are going to include a wide variety of bacteria. Some of these are going to be heterotrophs. So they're going to be feeding on other things. So they can be responsible for diseases. Also in the gamma proteobacteria, we find, interestingly, if you remember back in the days of Bio 211, Pseudomonas fluorescens, and the strain we studied, L5196. That is an example of a gamma proteobacteria. Here we have also delta proteobacteria. The example shown is an interesting one. This type of a myxobacterium shown here looks like a little tree with some kind of a fruit up at the top. Indeed, we call those fruiting bodies. And these are bacteria that in harsh conditions, they can actually attack other bacteria and take the nutrients and take the resources from other bacteria, effectively destroying the bacteria they attack. In the epsilon proteobacteria, one of the examples we're given is going to be H. pylori. H, or Helicobacter pylori, is the bacterium associated with stomach ulcers. In some places, people still believe that ulcers are the result of hydrochloric acid produced in the stomach for the purpose of digestion, and scientists for a long time have demonstrated that stomach ulcers are not the result of hydrochloric acid. Instead, they are the result of this bacterium you see featured here, colored red, at the bottom right corner of your screen. Those bacteria are very difficult to get rid of. And so in many parts of the world where the bacterium is found in water, people drink the water and they kind of like develop a permanent association with H. pylori. So they never really get very sick, but yet on occasion they can suffer discomfort caused by mild ulcers that they can develop. In extreme cases, these ulcers can become bleeding, and it can be a serious problem of the gastrointestinal system for people who suffer from this kind of an infection. But they can be treated. These bacteria can be destroyed with powerful antibiotics. I'm going to escape here. A second member of domain bacteria. So we've dealt with proteobacteria, gram-negative. Now we're going to be looking at chlamydia. Chlamydia are also going to be gram-negative. These are going to be different. They are going to be mostly parasites, live only within animal cells. And one of those classic examples of chlamydia bacteria are Chlamydia trachomatis. And this is responsible for non-gonococcal urethritis. That's an inflammation of the urethra. And this is something that is transmitted from one person to another through sexual intercourse. It is one of the most common sexually transmitted diseases around the world, and of course also here in the United States. Spirochetes, which now you can see in this photo, an example of a spirochete. These are going to be bacteria, gram-negative. They are going to have a helical shape, and the helical shape is going to be also the result of the flagella. These bacteria have flagella, but instead of projecting out of the cell, the flagellum kind of like wraps around the length of the cell and is surrounded by a membrane. And so that is a detail that is different from other bacteria, such as, I'm trying to think of the name, Salmonella. Salmonella has flagella, but those flagella are free. They are clear from the cell wall and the cell membrane. And so in this case, with this flagellum wrapped inside of the outermost membrane of these bacteria, it makes for like a snake-like motion. You may remember from Bio 212 those very small roundworms, nematodes, and how they have a snake-like type of motion. That is exactly the motion you can also see with these spirochetes. They are responsible for some serious diseases. Borrelia is one of those examples. Borrelia causes a type of disease known as Lyme disease, and it's transmitted from ticks to humans through tick bites, and is one of those bacteria that once inside of the body, it has proven very difficult to remove, very difficult to eradicate. Don't know what happened here, but I'm going to bring back my slideshow to slide presentation mode, and here we are. Okay. Oh. I'm sorry about my technical difficulties I'm having with my PowerPoint presentation. Okay, here we go again. Fourth group of bacteria is going to be cyanobacteria, and these we see in lab. The example we are shown in this illustration is Oscillatoria. This is a filamentous type of cyanobacterium. The appearance is like a filament because what we have here is actually many bacteria attached end-to-end, 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 making these long filaments. These are photosynthetic because they can use light and pigments such as chlorophyll to be able to produce their own sugar molecules. They produce oxygen as a byproduct, and this oxygen is one that we can benefit from, the type of oxygen we need for our own cellular respiration. And one last group we can make reference to when it comes to bacterial diversity is going to be the gram-positive bacteria. These are going to include many examples you may be familiar with. For example, Bacillus anthracis. We've seen this in lab and how these cells are capable of producing endospores to survive harsh environmental conditions. Clostridium botulinum. This has been traditionally associated with a severe type of food poisoning, what can result in death. It induces paralysis of the nervous system, and now scientists have figured out a way of extracting the botulinum toxin and use it for other treatments, including getting rid of aging lines in the faces of people. You've heard about Botox, for example, I'm sure, and other applications that the botulinum toxin now has in medicine. Staphylococcus is going to be a bacterium that lives on the skin of every human on Earth. Now, there are two kinds of Staphylococcus. There's aureus and Staphylococcus epidermidis. One of those, Staphylococcus aureus, is responsible for skin infections. And so if you ever suffer from a cut or a scrape and that became infected, that is because you have Staphylococcus aureus living on your skin. Mycoplasms are going to be the smallest known cells. And here you see a fibroblast. This is a special cell of the connective tissues of humans. And you can see the white dots on this fibroblast. The little white dots you see here are going to be precisely the little mycoplasms. And so that gives you an idea for the diversity of bacteria. And for sure, these groups we've mentioned today in this video are groups worth remembering and that I expect you to learn as part of the objectives for this class. That is all for right now. I'm going to make another video that deals with the ecology and ecological impact of bacteria, and that's going to be next. I'll see you soon. Goodbye.